the aim of this work was to really look at real-world allogeneic hematopoietic stem cell transplantation in a way that we've never been able to before, answering questions about how many of the transplanted cells actually engraft and survive in recipients long-term. Even though we've been doing clinical transplantation for almost half a century now, we still don't know those fundamental questions. Like, we know that we need to put 2 million CD34 positive cells per kilo of recipient body weight. How many of those are actually stem cells that survive and engraft through transplantation? And what happens, what are the consequences for that transplanted blood system? Does it aid the transplanted blood system? Does it acquire genomic damage? What happens to the clonal makeup of the transplanted system? So these are questions that we all wanted to look at through the study. We've used an approach that we've used in a number of studies at the Sanger, where it's based on the idea that every cell in your body acquires new somatic mutations from the first cell of the zygote after conception. Such that by the time you're about 60, each of your blood stem cells will have around 1,000 mutations. And you can use if you know the somatic mutations in single cells, multiple single cells from an individual, you can use the pattern of sharing to infer the family tree of those cells. And if you know the family tree, there's actually really powerful techniques that you can use to work out what's happened to that population through time. So we've used that approach previously to look at normal aging in blood and to look at some diseases such as myeloproliferative diseases. But in this study, we had transplant recipients and donors who were decades after the transplant procedure itself. And we were basically able to grow up single blood stem cell colonies from the donors and the recipients, sequence those, and to build a family tree of the donor blood system that has the same donor blood system in the donor and the recipient, but has simply diverged at the time of transplant. And from that, you can see what's happened to the transplanted blood system relative to how it's been in the donor, unperturbed through life, and therefore to kind of work out what the effect on the transplanted blood system is. The first thing that we've managed to find out is the first time have estimates for how many of the transplanted cells are surviving. And we found that in most cases, this figure is around 30,000, particularly when there's a young donor with a good number of CD34 positive cells that have been transplanted. But we found that if you have a lower transplanted stem cell dose, or the donor is older, on average, those individuals will have fewer stem cells that are actually surviving into the recipient. We've also found that the diversity of the clonal makeup in recipients is lower compared to donors. And we know that with age, your clonal diversity decreases. So this decrease in recipients corresponds to about 8 to 10 years of aging, roughly. Surprisingly, though, we found that the effects on mutations in recipients wasn't particularly significant, i.e. the hematopoietic stem cells in recipients had very few additional mutations, if any, compared to their donors. So that's quite reassuring, but it did have an effect on other features of aging on the recipient blood system, mainly in terms of clonal diversity. In some ways, this is quite early stages for this approach. We were able to apply it to 10 individuals, almost as a proof of principle, and to work out some of these fundamental parameters for the first time, to really work out how we can use it to optimize the transplant procedure. I think we'll need to do it in more individuals, so that will be a bit more work. But I think in the long term, this approach will be a gold standard to really understand what's going on in transplant with different parameters, different ages of the donor, different conditioning regimens, different types of stem cell source, to understand how these impact the fundamental parameters of a transplant. The other thing I wanted to mention, which I think is quite exciting, is that one thing that our research suggests is that some pre-existing clones in the donor are more likely to thrive through the transplantation procedure. And I think if we find changes in those cells that aren't a concern in terms of predisposing to cancer, for example, these could guide ways of enhancing the transplantation procedure. If we can mimic the changes in those clones, perhaps by some sort of drug, could we enhance transplantation more broadly and therefore make transplantation procedures a more successful, viable approach more broadly.